Good morning, and let me just begin my lecture, or two lectures, by saying that I welcome questions during the lecture. So if there's something that I say that's not clear, please interrupt me, and I'm happy to engage in a discussion. So the topic that I'm going to address this morning revolves around biological applications of little crystals. And as the title indicates, this is sort of distilled into the design of interfaces between synthetic materials and biological systems. So what I wanted to do at the beginning of my lecture is just try to put this work in a little bit broader context and tell you that this topic, not involving little crystals, but materials in general, is one that's received enormous attention over the past few decades. And so I wanted to address the question, well, why? And so why has there been a substantial interest in design of interfaces between synthetic and biological systems? And the answer is because the technological impact of such interfaces is very large. And one can describe it really from two perspectives. And so one is the question of how do you define or design synthetic materials that will provide cues to biological systems to direct their behaviours. And here as an example, what I want to tell you is that if you take a certain type of cell, a so-called cardiomyocyte, so it's the cell that comprises a heart, and if you put it into the right mechanical environment, and that you can do by defining, for example, the structure and properties of a polymer, you can cue a population of those cardiomyocytes to spontaneously beat in a synchronous fashion. And so that's an example where the properties of a synthetic system can cue a biological system into a useful behavior, and there's a large body of work going on looking at how to create artificial parts based on culture of cardiomyocytes in particular synthetic environments. So that's one perspective, and then the second is to use materials to tell you about biological systems. And so here, the idea would be to create materials, for example, that will respond to particular components, or, for example, mechanical forces developed by biological systems. And so there's a great deal of interest in creating sensors that can detect quickly the emerging diseases. And as the population becomes more mobile, and we jump on planes almost every week and fly between continents, so there's a redistribution of disease going on around the world. So to have diagnostics that can rapidly detect, for example, viruses, is something that would be very useful. And so this is also a driver of a good deal of work that revolves around this idea of biological interfaces. And so if you look into the literature, and you ask what approaches have people explored over the past few decades to create these interfaces between synthetic materials and biological systems, what you'll find is that there's an enormous diversity of materials. And I'm summarizing a few here. And so what I'm showing up here is the idea that if you control the topography of materials, that alone can have a dramatic impact on the behaviour of biological systems. And there's some very nice work done in Scotland by Curtis and Wilkinson showing that if you create topography and materials on the scale of hundreds of nanometers, you can cue living cells to exhibit certain types of behaviours which are reminiscent of their native in vivo behaviour. And the idea is that this length scale is similar to that which is associated with the extracellular matrix. And so the topography of materials is very important. And that includes topography over a wide range of length scales. So work done at my home institution, University of Wisconsin, by Sean Palatek, has shown that if you create topography of materials on a scale of tens of micrometers, so large compared to the length scale of a typical mammalian cell, which would be about 10 micrometers, then you can culture cells in ways that the cell quickly defines the microenvironment, and so cells secrete soluble factors, and you can build up concentrations of those factors in these microenvironments, and that feeds back into the cell behavior. And this particular image is showing some human embryonic stem cells being cultured, and if you want to differentiate them, you can achieve very nicely controlled differentiation in certain contexts in these types of microenvironments. And so a few other examples, of course, controlling the presentation of biological binding groups at surfaces is a very important area of ongoing activity. And so here it's showing short strands of DNA that will hybridize and so bind specifically with a complementary sequence, and so there are a lot of diagnostic devices based on these sorts of material interfaces. And then the last one I'll mention down here are hydrogels. And so these are polymeric materials that swell in the presence of water, and they might be 90% water and 10% polymer. And because of the aqueous environment, you can introduce proteins into hydrogels and they'll maintain their structure and function. And so a good deal of work is ongoing with hydrogels in the context of biological applications. And so this list could be a very long list, and so this is just to sort of illustrate what's going on. And what you'll notice here is that, and if you look into the literature, you'll find that despite the fact that a wide range of materials have been explored, what's surprisingly absent from the list are liquid crystalline materials. And so that's the topic that I want to address today. And so while it's been somewhat absent from design of synthetic materials, so in this biological application context, if you look at biological systems, liquid crystallinity is very common. And so if you take, for example, a dispersion of DNA and concentrate it, it will exhibit these beautiful interference colors that are characteristic of a cholesteric liquid crystalline phase. If you take a concentrated dispersion of virus particles, and so virus particles, some viruses are anisommetric, so they're rod-shaped, things like tobacco mosaic virus. These systems, just because of excluded volume interactions, will exhibit long-range order at fractions of 10 or 20 weight%. And in fact, some of the physics underlying so the understanding of the role of excluded volume interactions emerged from studies of virus particles, and so they're very useful to mesoscopic building blocks with which to explore the physics of liquid crystallinity. And then finally, what's over here is a depiction of a cell membrane, and so this is a phospholipid bilayer, and either protruding through it or attached to its surfaces are proteins and sugars. And what nature has worked out is that the liquid crystalline characteristics of a biological membrane can be used to advantage, and a lot of the useful properties of biological membranes, and I'll come back to that a little bit later in my lecture, are derived from this combination of order yet mobility that goes in hand with the liquid crystalline phase. And so the message here is that on the biological side of the interface, nature's worked out that liquid crystallinity is really useful. And so what I want to address is a biological side of the interface based on design of synthetic liquid crystals, and show that one can derive some useful behaviors from interfacing synthetic liquid crystals with biological systems. And so the generic question is, why? Why do it? And liquid crystals are exquisitely responsive materials, and in my lecture I'll talk about surface-driven anchoring transitions at biological And what I want to just sort of remind you of here is the characteristic energy scale associated with surface-driven ordering transitions, and that's 10 to minus 3 to 10 to minus 2 millijoules per meter squared. So one can, through subtle changes in the structure of an interface, change the energetics of interaction with the liquid crystal on this scale and drive ordering transitions in the liquid crystals. They're also responsive to very weak mechanical stresses, and typical elastic modulus of a liquid crystal is around 10 to the minus 11 newtons. And when cells attach to surfaces, they attach through these points of contact called focal adhesions. And a typical focal adhesion, you can just think of it as a point of attachment between a cell and a synthetic material, will transmit forces of the order of magnitude of nanonewtons. And there's an error here, that should be a micrometer squared. The typical area of a focal adhesion will be of order square micrometers. And so cells transmit to material stresses of the order of nanonewtons over micrometer square areas, and that's a sufficient stress to reorganize a low molecular weight liquid crystal. And so one can drive changes in the ordering of liquid crystals through forces or stresses of the magnitude that cells will generate. And so there's a very nice interplay between synthetic liquid crystals and cells based on mechanical interactions. And so just two other comments about liquid crystals from a very high level. Second is that in contrast to solid surfaces that I was referring to on the previous slide, the interfaces of low molecular weight liquid crystals are mobile. And so what this means is, just like a cell membrane, you can drive the reorganization of biomolecules at surfaces of liquid crystals. And this is something that I'll illustrate for you. You can use it to transduce biological interaction, or you can use the liquid crystal to drive the reorganization of biological molecules at interfaces. And then finally, if you want to use the liquid crystal as an actuator, you can actuate it using very weak fields. And so, of course, this is something that you do in a laptop computer display, is you apply a relatively weak field to drive the change in ordering of the liquid crystal, and one can do the same thing in sort of a biological context. And so the goal is not to change the optical appearance of the liquid crystal, but it's to change the behavior of the biological system through driving the liquid crystal.
So that this is the modulus of the Narcissus?
Yes. Yeah.
But the unit of modulus of the Narcissus is not needed. This is mechanical law, not modulus of the Narcissus.
Okay, so this is the order of magnitude of the elastic constant of
modulus of the Narcissus.
Yeah. Okay, so and the rest of my lecture I'm going to sort of divide it up into two parts. And the first is going to be something of a tutorial. And so I want to sort of point out to you the phenomena that enable biological applications of liquid crystals, and I want to talk through three of them, and one being surface-driven ordering transitions, phenomena involving the mechanical properties of liquid crystals, and then finally ionic phenomena. And a comment here is that if you're thinking about biological systems, you will have ions present, and they play a very important role in dictating the anchoring of liquid crystals at interfaces. And then in the second part of the lecture, what I'm going to do is combine the topics that I introduced here into an example, and that'll be to demonstrate the use of liquid crystals to detect the biological toxin that comes from gram-negative bacteria called endotoxin. Okay, so let's deal with the first topic of three and sort of the background. And so what I want to just to remind you is that many of the behaviors of liquid crystals in systems on the microscale really reflect a competition that involves these three phenomena. And so one that I've already introduced you to is the idea of surface anchoring, and so this is surface-induced orientation. And so I mentioned before that the energy per unit area that controls the preferred orientation of a liquid crystal at the surface is typically on this microjoule per meter squared scale. The second thing is the storage of energy in strained states of the liquid crystal, and in the simplest case, which actually is not always sufficient to describe the phenomena that I'll address a little bit later in the lecture, is this Frank free energy density, where you have three modes of strain, twist, bend, and splay. And as I mentioned earlier, so there's a characteristic modulus associated with each of these modes, and it's typically of this order of magnitude. And then finally, in confined systems, it's not always possible to accommodate the boundary conditions associated with the surface anchoring through a continuous strain of the liquid crystal, and this results in the introduction of discontinuities in the director of topological defects, and so those can play an important role in the systems that I'll describe too. And so if you take these two characteristic properties, these anchoring energies and the elastic modulus, in many of the systems that I'll describe to you this morning, what we're observing results from a competition between the two. And so you have a competition between surface anchoring of the liquid crystal on the surface and the elastic energy stored per unit volume in the strained state of the liquid crystal. And so you can ask at what length scale are those going to be comparable in magnitude, and it's given by the ratio of the elastic modulus to the anchoring energy. And so for these two values, that puts you in this 1 to 10 micrometer square area. And so the simplest one of the simplest sort of manifestations of that competition between elastic strain of the liquid crystal and surface anchoring can be seen in this so-called twisted pneumatic cell. And so, of course, this is the simplest form of a liquid crystal display, and so as you propagate polarized light through this twist distortion, linearly polarized light, its polarization is rotated, and if you apply an electric field, you can disrupt it. And so the torque balance that describes the orientational ordering of the liquid crystal in such a twisted cell is shown here, and so this is the torque associated with the twist of the liquid crystal, and this is the torque generated at the surface that we're considering at the top here, where the director, N sub E, is departing from the easy axis, so the lowest free energy orientation shown here. And so this departure from the easy axis involves work being performed, and the equilibrium orientation of the liquid crystal is going to be given by this sort of torque balance. And so this experiment is one that you can use to measure anchoring energies. And if you look at sort of the literature that leads to liquid crystal display, this type of measurement was commonly performed on things like polyimide surfaces, so surfaces of relatively simple chemical complexity. But what I want to do is that you can adapt this idea to biologically relevant surfaces. And so the first one that I want to talk about is a surface that's decorated with peptides. And so let me tell you why one might be interested in these types of peptides. And so what I'm showing here is a short peptide sequence. I think it contains about 12 amino acids. You'll notice that one of those amino acids is here, and it's called tyrosine. And so tyrosines are very important amino acid because it can be transformed, a so-called post-translational modification. So it just means a chemical transformation after the creation of the peptide or protein structure. And this particular chemical modification is the attachment of a phosphate group. And so this is what I'm depicting up here. And so here is the tyrosine, just showing the general sense. And here is the tyrosine after phosphorylation. And there is a set of enzymes that you will find in cells, and their job is to add phosphate groups, and so those are called kinases, and then there's another group which is to take away the phosphate group, and those are called phosphatases. And so what a cell is doing is it regulates protein functions by adding and subtracting phosphate groups. So when you add and subtract a phosphate group, you can induce conformational changes in the protein, and those result in changes in protein function. And in this particular case, the protein of interest is this transmembrane protein found in the cell membrane, and it's called the epidermal growth factor receptor. And this particular protein plays a key role in taking signals from outside of the cell and transmitting them to the nucleus, and the result being the proliferation of the cell. And so this particular protein is a key target for a whole range of anti-cancer agents. And the transfer of information through this intermediary, the EGFR, involves processes of post-translational modification, namely phosphorylation. And so there's a good deal of interest in developing methods to detect these sorts of transformations, and a lot of anti-cancer agents which are targeted at the EGFR are there to prevent this particular transformation. They're called tyrosine kinase inhibitors, and some of them form the basis of billion-dollar drugs. So the question is...
Another question,
yeah.
why is it important to get this group on? What does it mean for cell growth or relation to cancer, for example?
And so this particular protein, the EGFR, so when a ligand binds to it outside of the cell, what it does is it dimerizes, so you form two, and a consequence of that dimerization is the attachment of phosphate groups to tyrosines which are on the intracellular side of the membrane.
Also, it's in between the membrane.
Yes.
Okay.
And then when that phosphorylation occurs, you actually recruit other proteins to the surface of the EGFR, and that triggers an intracellular pathway involving the sequence of proteins, which ultimately sends a signal to the nucleus to promote cell division.
Okay, so...
And so when that signal link becomes dysregulated, you end up with excessive cell division.
Okay.
And so that's the, so the onset of cancer.
I have a question too.
Yeah.
So how do you prevent phosphorylation like that? I think it's threonine. Is that what it is?
So that's tyrosine.
No, OH. No, the
Yeah.
one, the other amino acid which has the
So this
OH
is,
group
yeah, that's the tyrosine.
on the left. See the other?
Oh, that's-
Not this one, the other one. Yeah,
Oh,
that
here.
Because wouldn't that also get phosphorylation?
So the enzyme, the kinase, it is specific in
For
its binding.
tyrosine?
Yeah.
For tyrosine? Okay.
And there are other kinases which will phosphorylate threonine.
Right, okay.
That's right. So there's threonine, serine,
Yeah.
and tyrosine B3, which will be phosphorylate. And so the question that we wanted to ask was, can we use anchoring transitions of liquid crystals to pick up subtle post-translational modifications such as phosphorylation? And so what we did was we immobilized these oligopeptides on surfaces, and we immobilized them in one of two states. And so one state is the phosphorylated state of the amino acid, and then the other state is the non-phosphorylated form. And so this is showing a measurement of the anchoring energy of a liquid crystal. And so this was really just a measure of the twisting of the liquid crystal. It's a super easy measurement to do. You just need a polarized light microscope. And this is the amount of peptide mobilized on the surface. And so this is increasing density of a mobilized peptide. And what I wanted to tell you is that indeed this sort of exquisite surface sensitivity of the ordering of the liquid crystal, which we're capturing in the sloping anchoring energy, is sufficiently sensitive to be able to distinguish between the phosphorylated state of this peptide and the non-phosphorylated state, which is the black triangles. And embedded in here is a second sort of control experiment where the tyrosine was substituted for phenylalanine just to explore what the role of this hydroxyl group was in the anchoring of the liquid crystal. And so we have a number of examples demonstrating that indeed one can use surface anchoring of liquid crystals to report subtle changes in the chemistry of biological molecules at surfaces. What I want to tell you is that extracting from the anchoring energy really the intermolecular interactions that result in these changes in anchoring energies is actually a very difficult challenge because of the chemical complexity of these interfaces. And so if you look at the diversity of chemical functional groups here compared to a polyimide surface, the workhorse of surface anchoring behaviors in the context of that kind of displays, you can see that things are far more challenging.
So if I get it right, so you go to the surface, for example glass, with one of these peptides, and you add the liquid crystal to see how much it sticks to the surface. That's simply said.
Yeah, and so the idea is that if you want to create an assay based on this, and so what you would do is you would either immobilize your peptide in a non-phosphorylated form, expose it, for example, to the epidermal growth factor receptor, and see to what extent you get phosphorylation of the peptide.
And under a microscope you see a difference in birefringence,
Yes.
optical readout.
Yes. Yeah, we see a change in the degree of rotation of the polarized light that's been transmitted through the molecules.
Explain?
Yeah.
So why is the anchoring is decreasing depending on the concentration of this technical stuff?
Yeah. That's because
Especially for this second stuff.
yeah. And so there's a lot behind this. So the question is, in the absence of the peptide on the surface, you see that you have this anchoring energy at around 4.5 microjoules. That should be microjoules, maybe this way. And that's because this substrate that we're using is a gold film that's been deposited by physical vapor deposition at an oblique angle incidence. And so that's actually the underlying substrate that is providing this anchoring energy.
Yes.
And then with increasing density of peptide on the surface, you're introducing intermolecular interactions that are in competition with the influence of the structure of that obliquely deposited gold.
Okay. Thank you.
Okay. And so one can go from what are biologically relatively well-defined and simple surfaces to complex surfaces, and I just wanted to sort of take a leap forward and tell you that we've been looking at detection of antibodies to emerging viruses, and this particular example is the West Nile virus, and for those of you from the US, you might remember that about 10 years ago, some veterinarians at a zoo in New York City, the Bronx Zoo, noticed that crows were dying. And that observation led to the realization that the West Nile virus had arrived in the United States. Actually, how it arrived there is not clear. But over the next 5 years, it slowly propagated across to the western states of the United States. And during that period, there weren't effective ways of detecting the presence of West Nile virus in the wide range of birds that were carrying the virus. And so we received some funding at that time to develop a diagnostic to antibodies to the West Nile virus that could be used for a variety of different animals. And this is just showing, this is surface-driven anchoring transitions. And so here the anchoring transition has been driven by the capture of an antibody at the surface. So a very complicated system. We're dealing with the serum of animals, which is a very tough topic because of all of the non-specific interactions that it can occur at surfaces. But the ideas can be translated. And so that's solid surfaces. And so if you're thinking about biological molecules, biology takes place in water. And so what I want to sort of introduce you to is a second type of surface at which we have been studying anchoring transitions. And it's one that I think is particularly promising in the sort of biological context. And so it's the aqueous liquid crystal interface. And so I'm thinking of thermotropic liquid crystals here. Of course, chromonics, and I think later in this workshop, you'll hear from Oleg Leventovich about chromonic liquid crystals. They are also very interesting water-based liquid crystals for biological applications. I want to focus on the interface between thermotropic and aqueous phases. And a typical thermotropic that I'll describe would be one formed, for example, from 5CV sodomatic liquid crystal. So how do you create the interface? So it's a little bit different, a little bit harder to stabilize the interface relative to a solid. And so what we typically do is we use capillary forces associated with microfabricated structures on surfaces, and the simplest way to sort of achieve the topography that leads to the capillary forces that will stabilize an aqueous liquid crystal interface is just to use an electron microscopy grid. And so you plop it down on a surface and you fill the pores of the grid with liquid crystal. And so this is the interface that we're interested in, the interface between the aqueous phase and this pneumatic. And so we're looking at a cross-sectional view of the grid. And on this silane treated glass slide, we've got homeotropic anchoring on the vertical surfaces of the metal grid, happens to be gold coated in this case. We also have homeotropic anchoring. And then at the aqueous liquid crystal interface, in the absence of any adsorbate there, one has planar anchoring, and so that gives rise to this sort of escaped radial type configuration of the liquid crystal. So minimization of the elastic.
So there's a mixture between like 5CV and water?
Yeah. So, well, it's not a mixture in the sense that they're immiscible. And so think of it as an oil-water two-phase system. The oil is the liquid crystal.
And how do you know that the water doesn't go on the spot where the 5CV is?
So it doesn't go there because of the fact that the surfaces of this cavity are hydrophobic surfaces. Right? And so it's capillary forces that keep the liquid crystal in this sort of hydrophobic surface. I can also tell you that de-wetting of these structures is a problem, and I'll show you some experiments in a minute where we start adding surfactants and lipids, and that sometimes you run into experimental challenges associated with de-wetting. So what can we do at this interface? And so this is a movie, and so you're looking at polarized light being transmitted through a film of liquid crystal and then a crossed polar. And so this is underwater. The shaking was a student adding a few drops of surfactant to the water, and the surfactant was this cationic surfactant. It's an oil-water interface, and so the surfactant will absorb to the oil-water interface, and when it does, it will trigger an anchoring transition in the liquid crystal. And so you see that the liquid crystal is undergoing a change in optical appearance, and that's because this monolayer of surfactant is absorbing at the surface of the liquid crystal and is triggering an anchoring transition. Yeah.
So did you also treat your gold grid with the OTS?
No.
So then it's originally showed the homeotropic anchoring.
Yeah.
With the
Yeah.
5C.
So that's a interesting point. So gold is such a high energy surface that as soon as you expose it to the ambient, it collects adventitious adsorbates from the atmosphere and it becomes hydrophobic. And when it becomes hydrophobic, the observation is that it gives rise to homeotropic anchoring. When we deposit gold films in an electron beam evaporator under high vacuum, actually when it first comes out of the vacuum chamber, if you put 5 CV on it at that time, it will be planar anchoring. So there is adsorbates on the surface of the gold that gives rise to that. So this is the final state. And so the directed profile is shown over here, and so you have homeotropic anchoring at the surfactant decorated interface of the liquid crystal. And the reason it looks dark, of course, is now when light is transmitted through this film of liquid crystal, there's no change in this polarization. And you see these bright edges, and that's where the anchoring on the surface of the gold grids is perturbing the orientation of them. And so this is a very simple experiment, and it's one that can be generalized to biological amplifiers. And so I'm showing here the absorption now of a lipid that is reminiscent of a component of the cell membrane. And so now what we're doing is we're introducing into aqueous solution vesicles of phospholipid and fusing them with the surface of the liquid crystal. It triggers an anchoring transition reminiscent of that you saw with the cationic surfactant. And so what you end up with a monolayer of phospholipid, so 1/2 of a lipid bilayer, so the structure of the cell membrane, where the presence and organization of that lipid bilayer is coupled to the ordering of the liquid crystal. And one of the interesting sort of aspects of this monolayer is that these lipids are mobile, and so they can reorganize just in the same way that they would reorganize in a cell membrane. And you can't see, I guess, these slides with the lights on, they can dim. So we've measured the diffusion coefficient of the lipids at the liquid crystal aqueous interface. And the typical diffusion coefficient is order 10 to the minus 12 meters squared per second. And what I want to tell you is that that diffusion coefficient is very similar to that of a biological membrane. And so this is a very primitive mimic of a biological membrane, where the organization of the lipid component of the membrane is coupled to the liquid crystal. And so what one can then do is you can think about questions of, well, what happens if I introduce a protein into the aqueous phase that binds to the lipid component of the biological membrane? Can I report the reorganization of the components of that membrane? So the example I'll give you here very quickly is that of an enzyme. So the enzyme is phospholipase A2. So this enzyme is the enzyme that you find in snake venom and bee stings. And its job in that context is to break down cell membranes to allow the toxin of the bee sting or the snake venom to enter the cell. So the activity is shown here, and what it does is it just hydrolyzes an ester bond. And so in this phospholipid, you have these two esters, and phospholipase A2 catalyzes the hydrolysis of one of them, resulting in this so-called lysolipid and a fatty acid. And so can one see that type of enzymatic reaction at this primitive mimic of a cell membrane? And so this is now showing the orientational response of a film of liquid crystal that's decorated with phospholipid to the activity of phospholipase A2. And so this is an ordering transition driven by an enzymatic reaction. And so one is using the liquid crystal to amplify the activity of the enzyme that's occurring within a monolayer into the optical scale, and one can follow the kinetics, and also there's information here about the spatial organization of the enzyme and lipid at the interface during this enzymatic reaction. Okay, so the message that I sort of want to convey to you in this first part is really just that one can use anchoring transitions to report a range of phenomena involving biological molecular interfaces. And the surface sensitivity of the liquid crystal allows you to amplify events that occur at the molecular scale into the optical scale. So a very nice way of transducing biomolecular interactions. There's lots of phenomena that we have observed and published in the literature. One of the key challenges is sort of the elucidation really of the underlying intermolecular interactions. The complexity of these interfaces makes that process very difficult. And then finally, I've sort of introduced you to this aqueous liquid crystal interface, and its traits are very different from those of the much more widely studied solid liquid crystal surface, and that's associated with the mobility of species at the interface. And it's also a deformable interface. I didn't mention that previously. And this mobility will pop up again in what I'm going to say in a few minutes. And so in addition to our own work, I want to mention that a number of other groups are now looking at these sorts of interfaces in a biological context, including very nice work going on at the University of Colorado involving hybridization of DNA. University of Manchester looking at screening of proteases, and there's some work going on in Singapore looking at nanoparticle biological membrane interactions using this decorated surface of the liquid crystal as a primitive mimic of a biological membrane. So let me now move sort of to the second key point. I'm going to go a little bit faster in light of time. And that's to come back to the idea of the role of the mechanical properties of liquid crystals in the phenomena that I've just been describing to. And so the way in which I've described these anchoring transitions is that the liquid crystal reports biomolecular interactions occurring at an interface. And that's really an oversimplification. The liquid crystal is actually a participant in what's going on at the interface. And that involves the mechanical properties of the liquid crystal. So let me sort of explain to you what I mean. So if we go back to the case of the phospholipid absorbing to the surface of the liquid crystal. So I showed you before that if you decorate this aqueous liquid crystal interface with phospholipid, you can achieve homeotropic anchoring. And so this is a sequence of images just showing what happens at the interface as a function of time of incubation of the aqueous solution of phospholipid against the interface of the liquid crystal. And you go from this initial planar orientation to a final homeotropic orientation. What happens at intermediate times, and so you'll see that in contrast to the surfactant case, which underwent a continuous anchoring transition, what happens in this case is that there's the nucleation and growth of homeotropic domain. So it's a discontinuous transition in the case of the phospholipid. And so if we zoom into one of those intermediate time points and just look at the interface using a fluorescently labeled phospholipid, we see that the reason that you have domains is that the distribution of lipid on the surface is not homogeneous. And so these are domains, so these are two-dimensional domains on the interface that are lipid lean, surrounded by continuous regions which are lipid rich. And that distribution of lipid on the interface is reflected in the patterned orientations of the liquid crystal, which is what we were looking at on the previous slide. And you see that this lipid lean region of the interface gives rise to this domain of liquid crystal that has a planar anchoring at the LC aqueous interface, surrounded by a region which has homeotropic anchoring. And even these smaller domains, which are about 10 micrometers in size, are faithfully reproduced in the orientation of the liquid crystal. And so the question is why? So why do we see the presence of domains on the interface. And dilauryl phosphatidylcholine, the lipid that I've been referring to, at the surface of water, it's been exhaustively studied, and it doesn't show coexistence of two phases at room temperature. And so it was a little bit of a surprise that we were seeing what looked like phase separation at the liquid crystal aqueous interface. And so that caused us to sort of ask what is the role of the liquid crystal in this phase separation? And so this is an experiment where we take advantage of the fact that 5Cb undergoes an aromatic to isotropic phase transition at about 34 degrees Celsius. And so what I'm showing up here is the fluorescent micrograph of a interface of the liquid crystal. So we're looking at the lipid here, and you see the presence of domains at 32 degrees, where 5Cb is in the active state. And you see these patterned orientations of the liquid crystal. So this is a polarized light micrograph. You heat the system up to 2 degrees or 2.5 degrees into the isotropic phase of 5Cb, and the domains disappear. You cool the system back down into the matic state. So you're going through bulk phase transitions between isotropic and the matic states. And on the interface, you're going between a homogeneous surface phase and a phase separated state on the interface. And so there's a coupling that's occurring between the liquid crystal and the phase behavior of the liquid on the interface. So it's not, the liquid crystal is not just a passive observer of what's going on this interface. So why is that? And so the elasticity of the liquid crystal is driving the phase separation of the lipid on the interface. And so in the initial state of the system, we have planar anchoring at the aqueous liquid crystal interface, and we have perpendicular anchoring at the solid liquid crystal interface. So we have splay and bend strain in the initial state. In the final state, we have released that strain of the liquid crystal because of a change in the easy axis of the liquid crystal at the lipid decorated interface of the liquid crystal. So at intermediate coverages of lipid, the system reorganizes the lipid at the interface into clusters that result in a change in the easy axis locally, and those release the elastic strain of the liquid crystal. And so the clustering of lipid is driven by the elasticity of the liquid crystal. And this sort of very simple idea can be captured in a primitive model. And so this is a model of the free energy of this lipid-decorated film of liquid crystal. And so we have the entropy of mixing of the lipid on the surface. We consider pairwise interactions between lipid molecules on the surface. And then we have two additional terms. We have a term that describes the elasticity of the elastic energy in the strained state of the liquid crystal, and then a surface anchoring term that comes about because of the departure of the director from the easy axis of the liquid crystal on the interface. So if you plot that free energy versus surface fraction of lipid, that's what this line here is. You see that you can introduce a point of double tangency. That's the phase separation of the lipid on the surface, and you can use that to construct a binodal. So this is temperature versus surface fraction of lipid, and you see that you have the coexistence of lipid-rich and lipid-thin phases on the surface. And so the phase behavior of this lipid within the monolayer can be manipulated by changing the elastic energy stored in the liquid crystal. So that elastic energy goes as 1 over the thickness of the film of liquid crystal. So you'd expect then the phase behavior to become a function of the thickness of the film, and indeed that's what you see. So you can get grids of different thickness, vary the thickness of the film from 5 to 40 micrometers. At 5 micrometers, you see that you have the phase separation. At 40 micrometers, you have a homogeneous surface phase of the liquid crystal. And so this idea that the pneumatic stresses can reorganize the biological molecules on the surface is actually a very useful one. And so you might have noticed in that movie I showed you of the activity of phospholipase A2, the anchoring transition that occurred was not homogeneous at all, but you saw domains forming on the interface. And so those domains are caused by the elasticity of the liquid crystal. And so they sweep and reorganize the lipid on the interface. And that actually allows us to detect the presence of the enzyme activity at very low concentrations. And so we see the appearance of domains using concentrations of enzymes which are actually down in the femtomolar range. And so the comment here is that the elasticity of the liquid crystal is very useful in revealing the activity of biological molecules at these interfaces by reorganizing them. And so that's really the second part of my message, is that the liquid crystal is not passive at all, but it's an active player in what's going on at the interface, and it can be used to drive the organization of these biological molecules at interfaces. And what I want to also tell you is that this idea that the mechanical stress in the film of liquid crystal can be used to manipulate the organization of the biological adsorbate is actually one that you find in biological systems. And so in bacteria, for example, bacteria use strain induced through the curvature of biological membranes to achieve local targeting of lipids that have architectures that accommodate that curvature. And so this is minimization of elastic energy, leading to spatial targeting of lipids to localized regions in bacteria. And I don't know if you can see this, but these were some optical micrographs of the E. coli. And E. coli is a rod-shaped bacterium, and there's a stain added that preferentially accumulates with a lipid called cardiolipin, and it likes to go to regions of high curvature, and you see that it decorates the poles of the bacteria. And so the message here is that this idea of using mechanical stresses to drive the spatial organization of adsorbates that one can realize in these synthetic liquid crystalline systems can be found already in biological systems. And so we're just reinventing design principles that you find in cells. So the very last thing I wanted to is about ions. And so if you want to design interfaces at which you're going to either drive or transduce biomolecular interactions, and they involve biological molecules, you will have ions present. And ions play a key role in dictating what goes on at these interfaces. So in liquid crystal display technology, you spend a lot of time removing the ions from liquid crystals. You can't in biological systems. You have to learn to live with them and actually use them to your advantage. And so let me tell you a little bit about ionic phenomena. And so if you take just a non-polar oil, so an alkane, and disperse it as little droplets in water, the interface between the oil and water accumulates charge. And you can see that if you apply an electric field across that dispersion of oil droplets in water. And that gives rise to a measurement of so-called electrophoretic mobility. So you measure the rate of migration of the ions, and from that you can extract a so-called zeta potential. And so this is the electrical potential at the shear plane, the hydrodynamic shear plane associated with that migrating droplet in the aqueous solution. And so these are measurements of zeta potentials of xylene droplets dispersed in water as a function of pH. And what I want to do is that you see the substantial accumulation of charge reflected in these zeta potentials. So as you increase the pH, meaning you have more hydroxide ions around in solution, you see the zeta potential become increasingly negative. And this is because that there's a preferential accumulation of negatively charged species at oil-water interfaces.
So I guess you use a DC field, or an AC field as an AC field?
That's correct.
What if you use an AC field with a low frequency and you still...
Depends on the frequency. So So this is a DC. If you go to sufficiently high frequency, then the ions won't have time to reorganize. So
You know, do you have an idea, guess about what you might do at the frequency?
I don't know. I guess
or less?
my guess is that it's above kilohertz. It's probably around 20 kilohertz and megahertz. So millisecond to microsecond timescale would be where you lose the electrophoretic mobility. Okay, and so the question actually is, why does this occur? And it's actually an unresolved mystery. So why do you have charging of oil-water interfaces? And the hypothesis is that there's preferential absorption of hydroxide ions. Why would a hydroxide ion want to go to the interface between a low dielectric medium and water? That is something which is not yet fully understood. But what is clear is that this accumulation of charge occurs, and it also occurs at the interface of the thermotropic lipid crystal. And so this is a zeta potential measurement of the interface between droplets of 5Cb and an aqueous electrolyte solution, so it's 100 millimoles sodium chloride. And you see also that you have increasingly negative zeta potentials. So potentials of minus 80 millimoles. So these are relatively high potentials. So what does it do to the anchoring of liquid crystals? Well, associated with the accumulation of that charge is an electrical double layer. And so you have bound surface charge at the interface, and then you have diffuse compensating charge in the diffuse part of the double layer. And the electric fields associated with those diffuse parts of the double layer are sufficient to induce anchoring transitions at these interfaces. And you can see it here at pH 6. We have planar anchoring of the liquid crystal at the aqueous interface. If you go up to pH 12.8, we have a high concentration of hydroxide ions in solution, and we hypothesize at the interface. You see that you flip to the homeotropic orientation. So why does this occur? Again, it's because of the electrical double layer. And so we have this accumulation of charge at the interface, and then on both sides of the interface, you have a concentration profile of the compensating counter ions. And we're familiar with the existence of those double layers in aqueous solutions, and so that's what I'm measuring when I measure the zeta potential. I'm measuring the potential at the hydrodynamic shear plane in that double layer. But what I'm describing in terms of the anchoring transition is actually a double layer that forms on the liquid crystal side of the interface. And so ions partition from the aqueous solution into the liquid crystal. You generate an electric field associated with the diffuse part of that double layer, and that acts on the dielectric anisotropy of the liquid crystal to hold the liquid crystal perpendicular to the interface. And so this is something that you can calculate, and you can't calculate many contributions to the anchoring energy, but this is one that you can.
Yeah, so in your cartoon, the interface between the liquid crystal and an aqueous solution is quite flat, but in the real case, I guess that it should have from the meniscus, right? Meniscus.
Yes.
So did you also consider that this meniscus effect?
Yeah, and we do. And part of the art of working with these grids is to fill them so that you don't have the meniscus.
Oh, okay.
And so you're right that if you overfill them, you will have a meniscus upward directed, and if you underfill them, it will be like this. And we can do that, and you can see that in the optical images that are generated by the liquid crystal. So we take a lot of care in actually creating these planar interfaces. And I'll, in a minute, talk a little bit about droplets. And so droplets, of course, you have curvature determined by the droplets. So you can solve the linearized Poisson-Boltzmann equation, and so this is describing the evolution of the electric potential as you move away from the bound charge at the surface due to the presence of the counter ions. And the key thing here is that the characteristic length scale over which the electric field decays at the interface is given by the Debye screening length, and that is dependent on the concentration of ions in solution. So once you have the profile of the electrical double layer that you solve from the Poisson-Boltzmann equation, you can calculate the contribution of that electric field to the anchoring energy. And so that's what we've done in this particular case. We have an aqueous solution containing 1 mole of salt. Some of that partitions into the liquid crystal, actually a relatively low concentration, because the self-energy of the ions goes as 1 over the dielectric constant, and the dielectric constant of the liquid crystal is much lower than that of water. But you end up with micromolar concentrations of ions. That gives rise to Debye screening lengths, which are typically in the tens of nanometers range. And if you use those Debye screening lengths to calculate the electrostatic contribution to the anchoring energy, it's of the microjoule per meter squared scale. So it's of the order of magnitude that should influence the anchoring of the liquid crystal. And indeed, if you start varying the pH of these aqueous solutions and the ionic strength, you can see changes in the anchoring of the liquid crystal, which are consistent with the influence of those electrical double layers on the anchoring energy.
Don't you have problems when you add salt to liquid crystal that you get some kind of phase transition? You get a change in the phase diagram, so you go to isotopic phase at the same temperature?
So that's right in principle, that's correct. These salts are partitioned into the liquid crystal at sufficient concentration to change the phase behavior. So these are micromolar concentrations. But if you go to much more polarizable or hydrophobic salts, like there are tetrabutyl ammonium salts, for example, which are quite hydrophobic, those you can partition into the liquid crystal at sufficient concentration that you will measurably change the phase behavior. So that's correct in principle. For these ones, the concentrations are sufficiently low that we have measured, for example, the nematic isotropic transition temperature, and it changes by less than 1/10 of a degree.
Okay.
So a very small perturbation. Okay. So I think in light of time, let me just sort of end by saying that not all ions are equal, and that there are also very interesting specific ion effects in these systems, and that's true of all biophysical phenomena. If you look at the solubility of proteins in salt solutions, it depends on the identity of the ions, and that's true of these anchoring phenomena. These so-called Hofmeister series that you may have heard of, there are Hofmeister series that control the anchoring behavior of the liquid crystal. So ions don't affect the liquid crystal just through electrical double layers, but there are additional ionic phenomena going on at these interfaces. And so in light of time, I think I'll just skip that. And so really the message here is that at these LC aqueous interfaces, ionic phenomena are important. And so when you're designing actuators or sensors based on liquid crystals, you have to think about controlling the ion content of a system, and you can control it to your advantage. And so this was the sort of end point of the first part of my lecture. And so the main points are summarized here. So the idea of using anchoring transitions to report biomolecular interactions, the aqueous liquid crystal interface as a particularly interesting class of LC interfaces for biological applications. The idea that the liquid crystal is not just a passive reporter, but it will change the organization of biological molecules at these interfaces. And then the importance of ions. So those are the key ideas. And so what I want to do to sort of end my